Hello, I'm Professor Vincent, and I'm glad you're interested enough in our research to watch this video. And I hope it's not too bad, and sorry that you're doing this online instead of being here in person to see this. So I'm going to share my screen here. And here we go. All right, I'm a bioinorganic chemist. That means I'm interested in molecules in life other than simple hydrocarbons and the derivatives. Most bioinorganic chemists are interested in transition metals because they have color or unpaired electrons that can be brought as spectroscopic probes to probe what's going on in a biomolecule. That is, most biomolecules that have metals use the metals as their central function. More than half of enzymes, for example, have metals in them, and the metals are the site of the catalysis. Now, this particular slide shows the elements that are essential for humans in red, those that have been proposed to be essential in yellow. Now, since the time this book was done, chromium shouldn't be shaded in anymore, and chromium is the focus of our work for the last 30 years. And it's our work in large part that led to that box being deshaded. All right. What we have shown is that the effect of chromium has been misinterpreted. What was thought to be a nutritional effect is a drug-like effect at very high doses. So chromium has been proposed to have beneficial effects on the symptoms of type 2 diabetes, and it does in at least rats, mice, and rabbits. All right, there it results in increased insulin sensitivity and lower bad and total cholesterol and lower triglyceride levels. Human studies haven't seen these results, largely because they're using about 10-fold too low a dose compared to what rats, mice, and rabbits get. All right, our research spans quite a gamut from inorganic synthesis and characterization through isolating and characterizing biomolecules, through cell culture work, all the way to working with animal models, doing toxicology and pharmacology. Right. In terms of our synthetic inorganic work, compound on the screen is a trinuclear chromium compound that we have 6 patents for use as a nutritional supplement or as a potential drug. I'm going to show you where our research can go. That compound was used in a study with Zucker obese rats and healthy rats. These are the obese rats, which basically a big ball of fat with a rat stuck in them. And we gave this chromium compound to rats for 12 weeks. And here are the results in healthy rats. This is the standard diet, and these reflect diets with increasing amounts of the chromium compound. You have no effect on glucose, so you keep your normal healthy glucose levels, but your total cholesterol and your bad cholesterol goes down, your good cholesterol stays the same, but your levels of triglyceride go down. And this normal glucose that you can maintain, you can do with less insulin, indicating increased insulin sensitivity. All right, if we look at glycated hemoglobin, which is a measure of the average amount of glucose in the blood for 90 days, chromium doesn't do anything for the healthy rats over various times through the study. However, for the obese rats, it lowers the glucose, and for a strain of the obese rats that go on and develop full-blown type 2 diabetes, it really has an effect lowering blood glucose. You can see the effects on the symptoms of diabetes. Now, also a result of our work, and I'll show you a brief bit of that, the European Food Safety Authority has determined that chromium is not an essential element for Europe. Recently, the National Institutes of Health, Office of Dietary Supplements, put out their new position paper on chromium, and they said that it's not an essential element. And the official body that does this between the National Academy and the USDA hasn't spent the money in its regular review cycle yet to review all vitamins and minerals. I'm sure they'll come to the same conclusion. But in our big study, that was the big part of the evidence for this, we gave rats a diet that had the lowest amount of chromium ever given a rat, and they maintained their health status perfectly fine. We then also supplemented the diet with the normal amount of chromium in the USDA designed rat diet, and then supplemented with additional chromium. When the rats were injected with glucose, and one looks at how much insulin was required to eliminate the added blood glucose, it turns out it goes down and down and down as you add chromium. The trick is this corresponds to only 16 micrograms per kilogram of diet when we don't add any, which is equivalent to a human taking a nutritional supplement. And they're normal. And if you added chromium up and beyond that, only do you start to see statistically significant effects. So it takes enormous amounts of chromium. All right, we've proposed how chromium might work in this. Imagine that you eat a meal, your blood sugar levels go up, pancreas releases insulin. It binds to a protein called insulin receptor on the outer membrane of cells, which tells the cell to start turning on the thousands of activities it does in response to insulin. One of these is to move chromium into the cells using the molecule transferred in the bloodstream that normally transports iron. Chromium goes into the cell, binds to a little peptide called chromodulin, whose function is to take chromium and normally remove it from the cell. We propose, though, that when you have a lot of chromium supplementation, that you make enough of this, it stays around, it binds to the activated insulin receptor, locking it in its activated conformation for longer than normal, so that eventually a protease comes, cleaves up the insulin, the receptor relaxes, this comes off and gets flushed out. But the result is increased insulin signaling. Right, and here's an example of a way that we've looked at to test this, but we still haven't done it in intact rats, which is harder to do than one might think. This is looking at the amount of glucose uptake by isolated fat cells. Sorry, my phone went off there. Right. If you take these muscle cells and you store them overnight in high glucose, it turns out that chromium or the peptide or the combination have no effect. But when storing them overnight, you wipe out their ability to respond to insulin. So you add insulin. Insulin doesn't have an effect like it does on normal cells. So that if you add insulin and chromium or the peptide, there's no effect. But in the presence of insulin, if you add chromium and the complex together, it restores a lot of insulin's ability to activate glucose transport. All right, so one of our projects is looking at how chromium functions in the body like a drug, activating insulin. The other is looking at chromium-6 carcinogenicity. So chromate is an important environmental toxin. It's introduced there primarily through industrial waste. And it was a focus for a movie a few years ago by Aaron Brockovich featuring Julia Roberts. You want to look at the significance of this. And chromate looks like phosphate and sulfate to the body. So it gets transported through phosphate and sulfate transporters and gets put in close proximity to DNA. Chromium-6 is a potent oxidant, and biological reducers can readily reduce it. And when they do so, they make chromium-3 ultimately, which wants to be 6 coordinate and grabs onto any nearby oxygens and nitrogens, which can include DNA. And when it does so, it forms adducts to DNA, just it, or it might grab another molecule and bind to DNA, making a ternary complex. And these ternary complexes, excuse me, or the degree to which these ternary complexes form have been related to how carcinogenic chromium-6 is. Now, understanding how all this happens has been a problem because there aren't good spectroscopic probes for looking at chromium-3 at the concentration in a biological system. We've worked out how to do that with peptides and proteins, and now we're taking our expertise and applying it to DNA for the first time. Right, our first publication doing this was in Chem Biochem in 2020. It was named top 10% publication in the journal, and it was actually focused on the journal's cover. So our strategy is to take short segments of DNA that we control the sequence of and look at where DNA, or excuse me, where chromium binds to DNA in a controlled fashion and figure out using two-dimensional NMR and EPR exactly where the sites of chromium binding are. And it turns out that in these binary complexes of just chromium and DNA, chromium binds to N7 of the guanine bases. Chromium's also 3+ is also bound to several water molecules that hydrogen bond and hold it in near the guanine in the major groove of the DNA, where it doesn't affect the structure of the DNA much, but the binary complexes aren't carcinogenic. Now, we're now working on in a controlled fashion, specifically synthesizing ternary complexes so that we can characterize them at an atomic level and look at their effects on the structure of DNA. And then we'll take the little piece of DNA with these ternary adducts, put it into cellular DNA, and see what that does to cells and whether it can kick in cancer. All right, so our current studies in the lab are looking at the effects of chromium as a drug, and we're specifically looking at how chromium is released from transferrin right now so that it can be bound by chromodulin and affect insulin signaling. As I mentioned, we're looking at, looking at the carcinogenicity of chromate at a molecular level. And we've also been working with Dr. Cassidy and her group, looking at how chromium 3 complexes can be used to aid the sequencing of proteins and peptides by mass spectroscopy. In terms of myself and my group, this is my 30th year here at Alabama, where we've been averaging well more than 4 publications a year. In the last several years, I've always had at least 4 publications. We have 9 patents over this time. We've been funded by several federal agencies, National Science Foundation, National Institutes of Health, Department of Agriculture, Department of the Interior, Environmental Protection Agencies, some private agencies, American Heart Association, American Diabetes Association, for example, and also by private industry. During this time, I put out 14 PhD students. The 15 is going to graduate this summer. So that's one every 2 years. But with the graduation, I have positions in my group. So I'm really interested in recruiting students. If you have any interest in our work, feel free to contact me, ask for papers, be happy to email them to you, have a Zoom meeting with you. And I'll also send Dr. Bonazzoni a copy of the PowerPoint I used for this. All right, so thank you and enjoy everything while you're participating in this recruiting weekend.